So the ribosomal RNA is the third and last of the three RNAs we're going to discuss. Consisting of RNA nucleotides, it forms a globular shape. It's joined together with proteins to actually construct the ribosome. If you recall from our cell structure unit, the ribosome is an organelle that makes proteins, but it is also constructed in the nucleolus of the cell. And it is partly made up of rRNA and the other part is made up of proteins. rRNA is the most abundant RNA in our cells and in our tissue. I'd like for you to take a moment now and pause the video. On page 9, there are 21 review questions. Really great to review right now, going all the way back to Griffith and all of the scientists important in the discovery of genetic matter, DNA. Take a moment right now, pause the video. Please take about 4, at most 5 minutes to complete questions 1 through 21. Welcome back, checking our answers. The animal that Griffith worked with were mice. What happened when he injected the he-killed pneumonia and live harmless together was that the pneumonia DNA transformed, essentially meaning that the harmless bacteria transformed to become harmful. And they knew this because the mice died and we were able to extract harmful bacteria from the mice's blood. Number 3, describe the point of Hershey and Chase's experiment. They were really hunting to see what was the genetic material. Was it DNA or was it RNA? And through their research with bacteriophages, which is what they worked with on number 4, are viruses that infect bacteria, they determined that DNA is genetic matter. Moving on to number 5. The phosphate or phosphorus was radioactively labeled in the DNA molecule, which is found connected to the sugar of the DNA's phosphate backbone. Number 6. The sulfur was radioactively labeled in the protein. Number 7. Genes are subunits of what level of DNA? Well, genes are subunits of chromosomes. Number 8. Bases are subunits of what level of DNA? Well, since bases are parts of nucleotides, they would be the subunits of a nucleotide. Number 9, describing the complementary base pairing rule, offering also the bond that keeps them together, and where on the DNA are they found, on the sides or within? Well, the complementary base pair rules are adenine to thymine, guanine to cytosine. The bond that holds them together is hydrogen, and they're found on the inner portion, on the inside of the DNA molecule. Please take a moment right now and check your complementary strand with mine up there on number 10. And number 11, DNA codes for proteins. That is essentially how organisms are different, is really by what proteins their DNA sequence code for. The monomer for a protein is an amino acid. Number 12, great review. List 4 differences between DNA and RNA. Well, DNA never leaves the nucleus. That is its home and it will not ever leave out of the nuclear pores. DNA is also made up of thymine. It is double-stranded and the sugar present is deoxyribose. RNA will be able to leave the nucleus. Uracil is the nitrogen-containing base present in RNA, and the single-stranded structure is a characteristic of RNA, as well as the sugar being ribose. When and why does DNA replication happen? Well, it happens prior to cellular division, and it's so that the new cell can have an exact copy of DNA for its genetic instructions. 14. What RNA is involved in transcription? Well, that would be mRNA. 15. What RNA folds onto itself like a T shape? That would be transfer RNA or tRNA. 16. What RNA makes up a ribosome? That would be the ribosomal RNA, rRNA. 17. What RNA reads the code of DNA? Well, since this is the message that's going to leave the nucleus, it is messenger RNA. 18. What RNA needs to be protected after transcription before it goes into the harsh world of the cell? This would be mRNA, and that process or step is called post-transcriptional modifications. I'd take a moment right now and write that down, number 18, post-transcriptional modifications. Number 19, what RNA is bilingual and what languages does it speak? This is tRNA, and it is able to read the nucleic acid and convert it into a protein. Number 20, what RNA is involved in transcription and translation? Well, this would actually involve messenger RNA. That would be involved in both transcription, and tRNA and rRNA in translation. And the definition for transcription is when we take one gene of DNA and we make an mRNA molecule from it. Translation, on the other hand, is when we convert the mRNA strand and code into proteins. Now, we know we do this in the ribosomes, and also the language between here would be mRNA to actually amino acids. And tRNA is responsible for that translation step. Moving on to page 9, let's talk about transcription and translation. So the process of transcription in detail is involved with the DNA code being transcribed into mRNA. And when this occurs is when a protein is actually needed to be constructed. So what will happen is only the needed gene is going to be copied from DNA to mRNA. Now, all 46 chromosomes are not transcribed at the same time. Now, why would they not be transcribed? Well, in the process of transcription, we're going to make a protein, and we know that one gene can code for one protein. If we were to transcribe all 46 chromosomes, there would be a lot of unnecessary proteins constructed. That is why only the selected gene that produces the appropriate protein for the function that is needed to be performed is what is transcribed. Where this will occur is in the nucleus, and it happens in the nucleus because DNA is the master code and never ever leaves. So why we have to go through this process? Well, a gene on the DNA molecule is going to be copied into mRNA. And that message is later going to be converted into proteins. And we know that proteins really do just give the functionality to each of our cells. So let's look at the breakdown of this process. And once again, if I'm moving too quickly and you need to pause the video, by all means, feel free to stop the video at any time so you can catch up with all of your notes. Steps of transcription. So the very first thing that's going to occur is the chromosome with the needed gene will actually unwind, and those hydrogen bonds are going to break. Our RNA polymerase will attach to the region referred to as the promoter region, and the DNA template will be the guide for RNA polymerase. Now, RNA polymerase will bring RNA nucleotides found free-floating in the nucleus. Now, remember, this originally comes from the nutrients that we eat in our diet. And I just want to kind of recap the transcription or the RNA complementary base pairs are different than DNA. Remember, A pairs with U and G pairs with C. So G and C will always be together. They're like a married couple. Now, since your RNA molecule does not have thymine in it, uracil will replace. And one strand of DNA with the needed gene is copied into a single-stranded mRNA. This is done by RNA polymerase. And as the code is read, it's actually read in sequences of 3 bases at a time. These 3 bases, as mentioned earlier, are referred to as a codon. So a codon is actually carried in the message. So it's carried by messenger RNA. Following the codon, RNA polymerase reaches a region that's referred to as the termination signal. So this would be at the end of the gene. And this terminates the process. RNA polymerase will release from the DNA molecule, and a newly formed RNA molecule will be created. The original DNA will re-hydrogen bond together. A single-stranded mRNA separates and goes to prepare for that harsh environment in the cell. So we're all right now on page 10 in our note packet, and there's a example for you to transcribe on your sheet. The code reads AATGGCCCTATT. Go ahead and take the next moment and transcribe that DNA strand. So now that you've had a moment to transcribe that DNA strand, the result of your transcription should be UUACCG So what you've just made is actually an mRNA molecule in the process referred to as transcription. Now we can see that this molecule in red was the DNA, and this molecule in white is actually the mRNA molecule. Now, when examining this mRNA molecule, we can see that we can break the code up into codons. And so that would simply be done by taking 3 nucleotides at a time and separating them just like so. That will result in 4 codons. Now, each codon will code for a specific amino acid, and this code will be read in codons, 3 nucleotides at a time. Good job. Take a moment right now. I'd like for you to pause the video. You're going to go ahead and you're going to watch the transcription alone video. This is listed in the lesson for January 4th. So the transcription alone video is a really great reference for you guys to go back to see all of the involving parties within the process of transcription. I'd like for you to go ahead and pause the video. You're going to head over to the transcription alone video on YouTube. You can find this again on your January 4th lesson. Go ahead and click that video. It's a little shorter, but it's concise and very detailed on how this process of transcription occurs. So on page 10 in your note packet, we're right at that practice section. Let's do this practice together. And this is not in your note packet. I will get to the one in your note packet in just a second. So we know that this first molecule is DNA. We can see thymine present, and we're being asked to create RNA, specifically messenger RNA. So take a moment right now and transcribe this DNA strand. So you should have gotten UAG, CAU, UGA. And we can see that this transcription molecule has uracil in it, and I would go through and break these up into codons. Excellent. Okay, so now let's go to the example that's in your packet on page 10. The DNA molecule that's offered to you is this right here. I want you to pause the video and transcribe that DNA molecule. Go ahead and as you transcribe, cut them into the codons. Remember, that's 3 nucleotides and a slash. 3 nucleotides and a slash. Pause the video. And the product that you should have come up with is AUG, CGU, AGU, CGA, UAA. And I'd like to know how many bases you can calculate in this mRNA strand. Now, if you sit down and count them, you should count 15. Now, when we look at the codons, we can see that there's actually 5 codons within this molecule. And the DNA will actually code specifically for proteins. So in this example that we have here, we would actually have 5 amino acids that are going to be produced at the end of this process, because each codon will come along with 1 amino acid. Excellent job. So take a moment. We're going to test you guys one more time. You've got this DNA strand. I want you to take a moment, pause the video, create the transcript from this DNA strand. This is on page 10. Your transcript should have come out to be AUGCGU AGUCGAUAA. Cut them into codons.